Alright guys, so today we're going to talk about the scientific method. Now this is one of the most important things to learn, especially when you're starting off in science classes. The scientific method is used in all of the scientists, and it's the same method, and regardless of if you're doing a biology or a chemistry or a physics experiment, this is kind of the process that you're going to go through. Now this example here does okay. I like ones where it's more circular, because really the scientific method, there's no start and end point. It's a circular thing. You get results and then you ask more questions and you do more experiments to get more results and data, and then you ask another question, and it kind of we gain more knowledge and knowledge every time we kind of go around, and so it kind of like our knowledge is spiraling upwards almost, really, right? We go around and around, and we're gaining knowledge every time we do an experiment and ask more questions and get more data. So we're kind of increasing our knowledge as we go through this. So we'll do a quick overview and then we'll break down each of these parts. You start off, really it says asking questions, so really maybe I think first up here should be observations, really. Because really, like, how did science start? People saw something, they were curious about it, and they asked a question, right? They saw something going on in nature, they saw an animal climbing this tree maybe, and they're like, oh, I wonder why that animal is climbing this tree. So it starts off with some observations, and we ask some questions because we're curious, right? Scientists are very curious people. You'll do some background research, you'll kind of explore the topic, so maybe if you're like, oh, I wonder why that that bird, let's say we're looking at birds, then this type of bird always goes to this certain type of tree, and so you're like, oh, I wonder why that bird likes to go to maple trees instead of oak trees more. And so then you'll do some research and see, other people may have already asked some questions like that, and maybe they've already done some research, and so you see what other people have found out first. Then you construct your hypothesis. Now this is actually a tricky area for a lot of students when they first start, is constructing a good hypothesis. Now we could say a hypothesis is kind of like a lot of people say it's an educated guess or statement. It's kind of a proposed explanation about an observation, is what your lesson on K-12's website says. Think of it as a template. If you were going to make a hypothesis, you would make it like an if-then statement. So if something happens, or if I do something, then what will happen? And so the example that we'll use today, and an experiment that I'll use today to go through this, will be feeding mealworms different kinds of food to see how much they grow, essentially. And mealworms, they're just a type of worm. They're just very common in science classes because they're easy to experiment with. So I could say, if I feed my mealworms different types of food, maybe I'll do apples or potatoes, then the mealworms that eat the potatoes will gain more weight than the mealworms who eat the apples, or something like that. So I'm saying, if I do something, if I feed these mealworms different diets, then I'm making my prediction with it, then the mealworms that eat the potatoes will gain more mass, or whatever I think that it is. It's an educated kind of guess. So after we do our hypothesis, we'll test with an experiment, and that makes sense, I think, right? You're gonna actually do the experiment after you make your hypothesis. Okay, so now that I've tested my experiment, I get some results. So I need to analyze results and draw conclusions, and this can be anything. Like results could be doing statistical tests, so stats, and you'll learn more about those as you get higher up in the sciences. You'll do probably lots of statistical tests. And then you draw conclusions based on your results. So you get your data, those are your results. Conclusions are how you interpret your results. So that's an important distinction. So results are like your data. Data gathered from your experiments. Your conclusions are how you interpret that data. interpret the data. So there's your difference, right? The results would be like the numbers. The results would be like, in our experiment, the mealworms, the weights or the mass of the different mealworms. And then our conclusions would be, okay, we look at this and then I interpret my results and I see maybe, oh, the mealworms that ate the apples actually gained more weight or gained more mass. So then we can say, okay, results of the hypothesis, if we found out we were wrong, if it was false, if we found out it was false, then we'd have to think again, and then we'd maybe make another hypothesis, and then we'd go through again, we'd do another experiment to test it again. So we're kind of going in a circular fashion here. And then notice then we were a real scientist, we'd report our results, we would like write a paper on it, and maybe publish it in a scientific journal. For students, basically meaning you'll be doing, like writing a lab report, would be reporting your results. Now this says hypothesis is true. Cross this out. We can never, ever prove a hypothesis is 100% true. Can never prove, I'm going to abbreviate here, hypothesis is 100% true. Nothing in science we can say is 100% true. Even gravity, even the theory of gravity, we can't say is 100% true. A theory just means a hypothesis that's been supported by lots and lots and lots and lots of data, but there could always be some weird explanation that we don't know about, right? Just like it used to be true, saying that sarcastically, that the Earth was flat. It's obviously not. New scientific research and technology came out, and now we know that the Earth is not flat. So we never say we can prove a hypothesis is 100% true. We can only prove it false. Okay, so I want to go back and talk about these in a little bit more detail, or a couple things in a little bit more detail here. Experiments. When you're doing an experiment, you have variables. You're working with variables. 3 kinds of variables: independent, dependent, and control variables. So let's do independent variable. It's the variable that you control. So in our experiment, and it's with the if statement, so if I vary the food of mealworms between potatoes and an apple, that would be the independent variable. It's what you're changing. You're changing what type of food they get. So let's say what you vary. The then statement would be your dependent variable. So in our example, dependent variable, we said if we feed the mealworms different food, like potatoes or apples, then the mealworms who eat the potatoes will gain the most mass. So we'd say mass would be our dependent variable, the mass of the mealworms. Oops, there we go. Mass of the mealworms would be our dependent variable. So if independent variable, what you vary, their type of food, what you vary, oh, I didn't put here. So type, in our example, it would be type of food. Then the dependent variable, mass of the mealworms, which is what you measure. The dependent variable is what you measure. And our last type would be the control variables. And control variables are all the other things that you're trying not to change. Because when you're doing science, you only want to look at one variable at a time, right? If I was changing, in our example, if I was changing more than the type of food, I wouldn't know what was causing the mass of mealworms to change. So let's say I was changing the type of food and the temperature where the mealworms were living at. And let's say some of them happen to gain more mass than others. I wouldn't know if it was the type of food they were eating or if it was the temperature that they were at. So you only want to change, you only have one independent variable in every experiment. And all the rest of the variables you want to hold as constant as possible between all the different mealworms or all your different trials. You don't want to change anything. So your control variables would be all the variables you keep the same in your experiment. All variables you keep constant. So in our mealworm experiment, that would be like we're holding the temperature the same, the amount of light the different mealworms get the same, maybe the humidity, if we can, the amount of food they get should all be the same when we do any experiment. Okay, so we have our independent, dependent, and control variables. I think that will do it for this lesson on the scientific method.